Okay, so we're here to talk about whether or not CRISPR-Cas9 should be used in embryos. So I should state at this point that I am here as a PhD student, as a researcher, as a person who uses this technique in my day-to-day work. I was literally analysing data from using this system this morning before I came here today to talk to you about this. So I'm going to assume a relatively high level of basic biology background here. You all, I hope, understand that CRISPR-Cas9 is the name of a system given that was published 3 years ago and has sort of become an incredibly hot topic, as Sarah said, since that point. Because essentially, previous technologies have not really allowed us to modify DNA in any sort of controllable fashion. And if you imagine that a cell has all of its instructions in the form of DNA in the instruction manual, and those instructions actually make up everything that you are and include all of the instructions for that. The concept of being able to edit that in any way, whether it's within your body from birth by a mutation that occurs by accident, or whether it's by taking a cell out of your body, modifying it in addition and putting it back in, is a concept that we need to sort of address. So CRISPR is novel because it actually targets the DNA. This is why it's so controversial. Previous technologies in research labs have sort of focused on what I like to call the sort of photocopy of our DNA instruction manuals. So DNA, if you imagine it as a paper manual setup, gets photocopied and then is turned into proteins like that. And we sort of have always intervened at that level rather than actually changing what the actual level says then. So I don't know how much you know about the fact that this is now also the centre of a patent dispute, which is a thing worth noting in our debate today. And this weekend even, a rather controversial paper came out from a chap called Eric Lander on the subject. And this is a thing this is a problem that encompasses both science, basic science, and what we can and cannot do within a research lab. What pharmaceutical companies want to use it for and how they want to use it to modify the tools that they already have. And also medicine, which is sort of, I think, where we come in here, which is to do with whether or not we should be allowed to modify human DNA in embryos and sort of start modifying our germlines in that fashion. So I beg the question, should this even be a patented tool? That's a personal view of mine. I find it quite troubling that this could be a financial process, but it's something that we need to be aware of and also need to discuss as a collective on the whole, I'd say. Another question is, and this is sort of where there's the two parts of whether or not CRISPR modification of embryos should be allowed, is the case that Sarah alluded to of Kathy Nearcan that's been talked about by the HFEA at the moment, is to do with whether or not CRISPR should be allowed to be used for basic research purposes. And then there's the problem of whether or not we can do it sort of therapeutically, and those are two separate things. So I use CRISPR-Cas9 in my research to address research questions, to sort of dissect mechanisms of how genes interact with each other, and that's a sort of a technical tool. But obviously there is this application in human beings as well. The sort of take-home message I want you to get from me essentially is to do with how simple and easy this technology is to use. So this is how CRISPR has superseded any of the previous renditions of the technology, in that it takes a matter of days to produce a piece of DNA that carries the information for using this system on it, literally days. We've had some students in our lab produce vectors and pieces of DNA to use the system in under a week successfully. So the pace of this field is moving very, very fast. And the sort of the modifications that people in my field of research, for example, are introducing to this system is really revolutionizing how we can use these tools. So the prospects are piling up very, very swiftly, essentially. We can use CRISPR-Cas9 now both to identify locations of genes, to modify them, to remove them. We can use them to carry other proteins to areas of interest, which is very important in fields of epigenetics, for example. And So this sort of dichotomy of what is good for research and what is good for use in human beings and how we want to use it therapeutically is something that we really as a collective do genuinely need to think about. And I think they might warrant having different answers, because simply by being able to apply the research that we have conducted thus far to conduct further experiments in human embryos, for example, as Kathy is suggesting to do in the human embryos, is a different question, and in my opinion warrants a different answer potentially to whether or not we should be modifying human beings. And if anyone has any questions regarding the basic sort of summary of how the CRISPR system works as a technology, I'm more than happy to provide that in the question and answer session. But yeah, the soundbite for this, I suppose, is the fact: should we be doing this? Well, I would argue, why have we bothered with any of the research that we've conducted if not to use it to better humanity anyway? So, not saying that we need to plough ahead straight into human beings and start making modified babies in the next week or so, but I certainly think it's the aim of all of our research as a field of biology and biological research. And more importantly, I feel that it's important that people know that this is so easy to do that people are doing it and will be doing it, and unless we sort of demystify this and sort of address the issues regarding it and how we feel towards the prospect of modifying our germlines, modifying our future children, and the fact that we can't take that back once we start it, I think we're going to have a lot more issues on our hands than we're possibly prepared to take on. So there we go, that's sort of my take on this subject.
It's a great pleasure to be here today, and I love talking about controversial topics. As Sarah said, I've been working in fertility and genetics for 30 years, and as the slide says, we've been creating life. So I'm going to try and be very provocative and really give some food for thought, following on from some of the technical parts of CRISPR. So Louise Brown, I'm sure you all know, born in 1978. I'm sure many of you know people that have gone through IVF. This is my son. I've got 3 IVF children, and IVF has really revolutionised obviously fertility treatment globally. It's a multi-billion-dollar business. There are IVF clinics, private IVF clinics, in almost probably every country of the world. It's hugely coming up now in the developing world. So this is really creating life on a financial scale. Now, over my 30 years, we've had lots of very controversial things happen and lots of new technology brought in. And I just want to link things to do with genetics. So at the bottom here, we've got a picture of ICSI. So ICSI was first developed in 1992, and this procedure revolutionised male infertility. But there's some interesting caveats to that. We've got men that have a genetic or chromosomal abnormality that have been in the past totally infertile. They could never reproduce before this time. And now with ICSI, we can start them, we can now help them reproduce. And a classic example of this is cystic fibrosis men who are infertile because of congenital absence of the vas. So now with this ICSI technology, these men were totally azoospermic. They could never produce offspring in the past, and now with IVF technology we have helped these men that have a genetic abnormality to have children, and this will further with other new techniques that we're developing. Now we all know, Sarah's already mentioned the mitochondria at the top. We've got egg freezing, we've got lots of exciting things happening in IVF, but we're also, as I said, pushing back the boundaries. We've got now for male gay couples, without IVF technology they wouldn't have been able to have children. We've got Elton John here with his husband, and for men, gay men wanting to have children and go through egg donation and surrogacy, this is one of the most fastly growing areas of fertility treatment. So they have an egg donor and a surrogate to carry their child for them. And The Handmaid's Tale there, a fantastic book by Margaret Atwood, now coming into effect. There's a number of rich and famous that you all know that now have surrogacy. Why are they having surrogacy? We're using this technology to help them. And Hollywood actresses that are in their 50s or late 40s having twins because they've gone through this IVF technology. But one take-home message from this is that it is a class system. It's patients that can pay for this. Even in the UK, even when we have some funded IVF, the majority of patients going through IVF in the UK and globally pay for their treatment. So we have a class system, and that's something we really need to think about in relation to CRISPR. So as Sarah said, I've been working for many decades on pre-implantation genetic diagnosis, and from 1 or 2 cells from an embryo, we can now test this embryo for genetic disease. And we can now use next generation sequencing to actually sequence the chromosomes from these embryos. And it's not going to be long, very, very soon, before we can dig deeper into that data and actually get the full gene information of a pre-implantation embryo before we implant it. There are clinics in the USA now that are using this method just to look at chromosomes, but they're applying this to every patient and every single embryo. So not patients at risk of a genetic disease, which is what the procedure was designed for, but just every patient that comes through their clinic. So again, we're coming back to the finances. Who's going to be able to select their embryo for genetic characteristics that they feel from the embryos they produce are the genetically best embryo that them as a couple could make? And we've got Gattaca here, we've got Brave New World. So we've got reproductive and genetic classism. Now, many years ago I went to a lecture by Stephen Hawkins, and he said, Star Trek have got it wrong. In Star Trek, everyone looks the same as we do now. And he said very clearly, that's not going to be the case. We are going to genetically engineer ourselves to make ourselves better than we are now. For example, I'm very short. I'm sure if I genetically engineered my embryo, I would have made myself taller. There are disadvantages of being short, unless you're on an aeroplane. So we're very lucky in the UK. We have the HFEA, and we really lead with regulation globally. We are very well respected for discussing these topics and bringing them to the table. Now, one thing I just want to give you as my take-home message, yes, I'm sure we will bring in CRISPR. We love new technology. We all love the new iPhone, the electric car, all of these things. We are going to develop. This gives us a lot of interest and excitement in our field. But we have to think very carefully about new technology. We have to make sure that it's validated, that it's efficient, that it's not going to lead to any problems in future offspring, and that it's also ethically acceptable. And as I said, in the UK, we have forums to discuss these topics and to really lead globally in this field. And I've written 2 papers in this area. The first one's called When and How Should New Technology Be Brought into the IVF Lab? It will be brought in, we just have to do it, making sure that we've thought of all the possibilities and that it really is safe for our future generations. Thank you.
There's no legal answer, I'm afraid, to the question, should we be using CRISPR-Cas9 to experiment on human embryos? The law permits CC9 experimentation on human embryos, subject to an HFEA license, on the same basis as any other research application, but has nothing to say about whether we should. My personal view is that we should. To elaborate a little on the law, a successful application for research purposes was set out a research purpose, was satisfy the HFEA licensing committee that it has one or more principal purposes listed by the HFEA Act. CC9, that means CRISPR-Cas9, CC9 and other genomic editing tools are highly applicable to all of these, quotes, principal purposes. But subordinate purposes remain a concern to some. For example, incidental research into the viability of edited embryos for implantation purposes. Well, although gene editing of embryos destined for implantation, or the gametes used to make them, is not permissible, the position could change with very minor amendments to the law. The HFEA Act was amended 7 years ago to provide a framework for mitochondrial donation, under which are now the Mitochondrial Regulations 2015. Article 7 of those regulations permits third-party pronuclear transfer and maternal spindle transfer for the purpose of preventing the transmission of serious mitochondrial disease. It would be straightforward to add mitochondrial DNA editing to Article 7. There's a big difference between mitochondrial DNA and mitochondrial genes. Only 13 highly conserved proteins are coded by genes in mitochondria themselves, together with 24 genes for making them. The remaining 1,500 or so other mitochondrial genes are located in the nucleus. Why not edit them to prevent the transmission of mitochondrial disease too? And if these, why not other nuclear genes to prevent the transmission of other serious heritable diseases? Opponents of embryonic gene editing commonly praise two international conventions of 1997 in support of what they claim is the illegality of embryonic editing: the UNESCO Declaration on the Human Genome and the Council of Europe's Oviedo Convention on Human Rights and Biomedicine. The UK has subscribed to neither, but factual and semantic precision of the terms the human genome and the human germ line illustrates that these instruments do not necessarily forbid the editing of human inheritance, though they may in some instances. Okay, so Article 1 of the UNESCO Declaration makes an important statement of principle. It says that the human genome underlines the fundamental unity of all members of the human family, as well as the recognition of their inherent dignity and diversity. In a symbolic sense, it is the heritage of humanity. That's what Article 1 says. Well, no human has ever been born with, quotes, the human genome. Genomes belong to individuals, not populations. They're hybrids made from parts of parents' genomes arising from conception and expiring on death. Human reproduction fundamentally disrupts existing genomes, taking two and giving one. That, of course, is why we have sex. Amongst other reasons. The word diversity in that Article 1 confirms that the human genome refers to something more comprehensive, the set of all human genes in all conceivable human genomes. The UNESCO Declaration, in other words, is referring to humanity's enduring and evolving gene pool and not to individual mortal genomes. Turning to the other term that's often misconstrued, the human germ line. And the word line. Well, there's obviously no line from the father, we know that, but there's almost none from the mother. The 37 maternally inherited mitochondrial genes are the only genes that meaningfully, to avoid the hurly-burly of sex, to travel together and in an uninterrupted person-to-person line. The most rational meaning for the human germline, like the human genome, is understood at the level of the human population as a whole, not at the level of individuals. It refers to the totality of transmission within the entire human gene pool through all generations of humans and their antecedents. And this view is confirmed by the Biotechnology Directive, which, in contrast to the Oviedo Declaration and UNESCO Declaration, the UK is bound by. And this elegantly brings these two expressions together, these two ideas together, when prohibiting patenting. And it says that the patenting of process that forbids the patenting of, and I quote, processes for modifying the germline genetic identity of human beings. This is significant. Human-to-human mitochondrial transfer doesn't change the germline genetic identity of human beings, nor does replacement of a genetic mutation with a recognized working sequence from another human. On the other hand, some interventions would modify that identity, and we need to assess each of these coolly and rationally in the light of experimental evidence. For example, it may be possible to alter the germline inheritance, the germline identity of human beings without adding any new material, merely through deletions or sequence changes. Equally, germline genetic identity may not be affected by non-maternal differences. Okay. Okay. So I think I'll just quickly skip through. So variants inevitably occur. So I think probably what is your wrapping note? Okay, that's fine. Okay. So I'll just briefly say that the Oviedo Convention, which is the European Council Convention, effectively says the same thing. So I would suggest that actually there are differences to be made, distinctions to be drawn.
The question is, how do we monitor the off-target effects? Is that right? Right. Okay. Well, there's multiple ways. You can use bioinformatic tools to sort of predict essentially where you might be having to look for that, and then sequencing allows you to look specifically in those areas. That's for more of a low budget sort of lab. If this sort of research is actually condoned and allowed, and there's the parallel field of sequencing that's also developing alongside us at the moment. So sequencing is getting a lot easier. I can see quite easily in the future, when the cost of this comes down within the next couple of years, I mean in the future, not 10 years or so, it's going to be possible just simply to sequence the whole genome of said cell that's been modified, and you'll be able to assay the entire thing in its entirety. I mean, at the moment that's still possible, you can do that, but it costs quite a lot. So, you know, yes, that's the main limiting factor at the moment. But yeah, you sequence it, you look.
Okay, next question. Please introduce yourselves as well, if possible.
Alison Hall from the PhD Foundation. Sorry, I don't know why I'm looking for. Do the panel have any thoughts on what might be the first clinical applications of this technology? Particularly thinking about what Julian said about the example of the mitochondrial regulations and restricting the technology to serious disease.
Yep. So when we in our PGD program, we have occasionally had couples that both have an abnormality, both have a genetic abnormality, and we've done a few cycles for them. I know people globally have done this as well, and their chance of producing a normal child, because they're both carrying an abnormality, is really quite low. Theoretically, statistically, you should get normal embryos from them, but it's normally sort of Murphy's Law, and the embryos are affected with one or the other, or both. And there have been very, very few in our experience that have been. So I can imagine cases like that where both of the couple are carrying a genetic, or one of them might be carrying chromosomal and one carrying a genetic abnormality. But I've thought long and hard about why we would use it. So medically is one thing, but to enhance our population is obviously something that comes up ethically. So that's what I wanted to ask the audience. What do you think in our lifetime, some of you are quite young, so some of us are a bit older, but in our lifetime, do you think we would use this to enhance the population as Stephen Hawking thought we would? Show of hands, who thinks we would use this in our lifetime to enhance our population? If you had the money. Okay, a few.
And of those who put the hands up, what would you use it to enhance? What would you, Joyce has already said height.
Yeah, both a question and an answer. My question is for Joyce. You said we need to make sure we've thought of all the possibilities, but history suggests that's one thing we've never, ever managed with any other kind of advance, and I just worry about the optimism there. In answer to the question, I think, and I again throw this back to the panel, it seems to me absolutely inevitable that quite early on in the processes, scientists or particularly pressure groups, couples for instance, will be seeking enhancements, not purely sort of disease elimination and so on, but very, very early on, couples will be seeking enhancements, both particularly aesthetic ones, I think, but also intellectual capabilities and so on. So it seems to me that there is an inevitability that that's going to be happening, and therefore it seems to me that what we need to be talking about all the time is the regulation of that.
Yeah, I agree. I think it is inevitable. That's what I was trying to say from my talk. I am an optimist, so yes, I hope that we will discuss as much as we can within this. But even with PGD at the moment, PGD is used for sex selection, just for social reasons. It's apparently banned in the UK, but the technology we use now to look at chromosomes, you know the sex of the embryo. So if you've got a couple paying £8,000 or £10,000 for treatment, and you've got this genetic information, we have this information, and if they say, oh, I want a boy this time or a girl, obviously it does happen. It happens in the whole of Europe and globally, and this will increase. So as we get more and more genetic information, PGD is really the standing ground for the advancement of how CRISPR will develop. People already very soon with next generation sequencing are going to say, okay, I want the one with this colour eyes, or this the genes that might help sports ability or musical ability or whatever, and that will happen. That will definitely happen. They're paying for it. That's the issue. They are paying for it. It's not going to be open to everybody.
It won't be. Sorry, I don't know if this is on.
No, it's not.
Hello. I don't know if it works.
Sorry, yes, they probably will do that, but they won't be allowed to get that because currently under the mitochondrial regulations, there has to be the egg from the mother has to have actually been one which would have led to inevitability of a serious mitochondrial disease. So there has, if the idea is not to avoid serious mitochondrial disease, you don't get your ticket. This is licensed. Critically, the regulations do not say yes, you can do it. That's not how the system works or has ever worked. It is always, as we're seeing at the moment in relation to research, Catherine Milliken's application, it's always filtered through the licensing committee of the Human Fertilisation and Embryology Authority.
Can I just add one thing? So in fertility treatment, a big area of research is cross-border reproductive care. So if you can't have something done in your country, you just go to another country and have it done there. And we are the most highly regulated country. So in other countries, as I said, sex selections are being done in the UK, I assure you. But it's, as the technology moves forward in PGD and in CRISPR, you will just go to the country where it's allowed. So.
My point relates to that. I realise we've totally monopolised this discussion with the public right now, but that point is very valid in regards to research, actually. I mean, I'm sure most of you saw the news stories recently about the Chinese scientists who've started modifying humans and all that. The point about this is, like I was trying to say in my opening remarks, this is incredibly easy to do. Anybody with a molecular biology lab and basic molecular biology training and equipment can do this, theoretically. And it's not going to take very long. So this is actually to do with a technology that is available to everybody on this planet, basically, every single country, and how different societies and how different cultures approach this is going to affect and is affecting the kind of research that's being done. And there's always a disjoint between what researchers want to research and how they're researching it, and the tools that people like myself develop to address biological questions will always find novel applications in medicine to help further a sort of a clinical application as well. So we really do need to start thinking about this sort of disjoint and the pace at which this is going, and the fact that different countries will be doing it differently and are doing it differently, and how we want to sort of interact with that is really what everyone really needs to start thinking about, in my opinion.
We've only got 5 minutes left, so if you have some more questions, please.
Yes. Could you speak?
It was on. Oh, it is on. Hi, Jess Buxton, Progress Educational Trust. In answer to the question about enhancement, I think while there might be some demand for blonde hair, sporting ability, whatever, I think maybe also more pressing priorities might be, for example, wanting to enhance embryos to be resistant to infectious diseases, particularly given the fact that we're running out of antibiotics that work, Ebola, HIV, who wouldn't want to confer that kind of enhancement potentially on the next generation in many years' time, if it's all safe and the rest of it. And then sort of related to that, I guess the initial clinical indications are going to come from couples currently seeking PGD who don't have embryos that are healthy to implant, which are families at the moment who have a history of a particular genetic disease. So given that many genetic diseases, there's a high new mutation rate, for example, Duchenne muscular dystrophy, a third of cases I think are new mutations, so there's no family history. do you envisage and would you ever see a future where it might be seen as irresponsible to go ahead with reproduction the normal way and not kind of make sure your embryos are completely healthy before you even embark on pregnancy?
That's Gattaca. So my colleague Siobhan's in the crowd and she cringes every time I mention Gattaca. But that certainly is, and Brave New World, etc. You use the word safe when we know it's safe. I would say, just to be devil's advocate, we don't know anything we do is safe. How do we have any long-term studies? Years ago, I sat in a bar with Bob Edwards, the pioneer of IVF, and said, at that time, none of the IVF children had reproduced. I think it was about 15 years after IVF. I said, we don't know what we've really done. We don't know the long-term effects of the doses of fertility drugs we give the patients, or the ultrasounds, culturing the embryos in a culture media. We don't know the long-term effects of that. Every pill and potion that you take, we don't know the long-term effects. You know, my friends would say, oh, it's all been tested. We can't test what generations of an effect will have. So I would hesitate very much about using the word safe on any of the technology that we're doing.
I'm not going to say as a scientist. Yeah, knowing is a strong term. I think people often look at science and scientists and say, well, we want a yes/no answer. Is it safe? Is it not? None of this is a black and white situation. You don't know that life is safe at the best of times. And the point about science is it tests questions and provides essentially the strongest indications, and it's a prediction game, right? Not a game. This is not light matter, I understand that. But the point is, again, in relation to your question regarding immunity and that kind of sort of, could we modify our embryos to have stronger adaptive immune systems so that they are less likely to fall ill to resistant bacteria? We don't even know the basics of immunology yet to be able to do that. And so a massive part of being allowed to use the CRISPR-Cas9 system for research is to further improve our understanding of basic biology. And then the question then becomes, of course, once you know these things, do you apply them? But that's a very different, fundamental difference there that people really need to sort of be aware of.